Hello everyone and welcome to this Cytognos webinar co-hosted by BIOCOM, our partner in South Africa. My name is Arantxa Huarriz. I work as product specialist at Cytognos, and I am presenting today your cheat sheet for Smarter Clinical Flow Cytometry Testing webinar. If you have any question or whatever, you can write it on the chat, and then at the end of the webinar, I will go through all of your question or comments that you could have. Also, my colleague Samu from BIOCOM Africa will be monitoring the chat box, so if you have any question, you can write it, and we will try to answer you. Okay, so let's start. So these are the points that we are going to cover today. So, well, first I will start highlighting the value of the flow cytometry in clinical practice. Then I will talk about the current issues we face in the clinical routine lab. I will focus on the specific challenges of the minimal residual disease assays and on the design and validation of flow cytometry tests. Finally, we will see how we can overcome these challenges with a fully standardized solution, which is the EuroFlow method. And then at the end of the webinar, as I mentioned, we will have some minutes for any question or comment that you might have. So, well, let's start talking about the flow cytometry. And more specifically, the role of flow cytometry in immunophenotyping in the clinical lab, because it allows the diagnosis, but also the classification of leukemia, lymphoma, and plasma cell dyscrasias, and the detection of minimal residual disease after treatment. Flow cytometry is also used for studying the immune response of patients and for progress control of the treatment itself, and in the clinical immunology field to support primary immunodeficiency diagnosis. Therefore, flow cytometry is definitely a versatile and powerful technique with many applications in clinical setting, but it is also complex and it has multiple steps. For example, first, we need to collect the specimen, we need to transport it to the lab, and once we get it there, we have to process the sample and we have to stain it. Then we will need to acquire the tubes in the instrument which has been previously calibrated and compensated. And last step is analyzing the data and reporting the results to the clinician. So as you can see and you know, it is a long process. There are multiple checkpoints here where different errors may be introduced and ignored, especially in the pre-analytical and post-analytical phase. And these errors have economical consequences, but more important, negative effects in the patient care. That's why all flow cytometry assays require careful assay design, instrument setup, performance validation, and ongoing quality monitoring. Okay, so now we are going to see the most common issues we face in clinical routine briefly, and then finally how we can overcome them. So first, as we just mentioned, sample stability can be an issue because, well, most of the time we require a fresh sample, in some cases no longer than 36 hours after collection, because then the percentage of debris increase. Also, the samples that come into the lab are very different. We can work with peripheral blood, with bone marrow, lymph nodes, etc. And they come with different anticoagulants like EDTA, heparin, etc. So they will require different shipping, storage conditions, processing time, etc. And we have to take into account to perform the correct processing. In regard to the second step, there are different WASH protocols in place, so we have to coordinate multiple workflow at the same time and apply the corresponding one depending on the sample or depending on the study that we are performing. Regarding the sample staining, cocktailing the single antibodies can lead to pipetting errors. Also, if we get a new antibody lot, we need to cross-check it with the previous one. And we need to have an efficient control stock of all the reagent to reduce reagent waste. For the machines, our instruments have to be correctly set up, and we need to track day-to-day performance to ensure consistent data over time and obtain reliable and reproducible data. In addition, the optical alignment, fluidics, laser power, and photomultiplier voltages must be also monitored. It is also important to acquire the tubes as soon as possible to minimize sedimentation and not to compromise the quality of the sample. But also then, during acquisition, we can run into issues such as unstable flow, clogging, or air bubbles that could affect on the results. When it comes to the analysis, there is always a user bias because positive and negative population are clear or can be clear, but there is a lot of middle ground in between. So the final results highly depend on the analyst expertise. Lastly, we have to put together a report with our lab test results, transfer it to the LIST system so it can be accessible to the pathologists and clinicians. And of course, ensuring that all the sensitive data is protected. Okay, so those are common points, common issues regardless of the type of study, challenges that we can find in any lab. However, more sophisticated tests, such as minimal residual disease assays, pose additional challenges. So let's take a look over them. Now, to follow a patient, we usually analyze bone marrow samples, which is a very delicate specimen. For example, we only get a small volume to run the test. Or we can have hemodilution issues if we don't get the first pool aspirate that will affect the result. Moreover, the anticoagulant may affect marker expression. For example, it has been proved that heparin that regulates CD138 expression in plasma cells. In addition, when performing these studies, we need to achieve a very high sensitivity, and for that we have to bullize the sample up front to concentrate the number of cells, so we need to add an extra step to our sample processing protocol. We need as well to make sure that our minimal residual disease panels are optimized for detecting the target population at very low levels, and that they are adapted to the immunotherapies that can interfere with expression of certain markers, as in the case of anti-CD38 treatment in multiple myeloma MRD, because the plasma cells can be affected by this treatment. And definitely, we need to make sure that our instrument has the capacity to collect a high number of events relatively fast. One of the biggest challenges in minimal residual disease assays are the false minimal residual disease negatives. In these studies, we are handling a large number of events, and we want to identify a population that has a very low frequency. So high sensitivity is a key to avoid reporting false MRD negatives. Also, we need to consider the patient treatment for the gating strategy, as some immunotherapies can mask or can affect the expression of certain antibodies, as we mentioned. On top of that, in every pathology lab, we are getting more and more cases. We have a very tight TAT, and in addition, because of the COVID-19 pandemic, the hospitals may have cut the budget for the lab. And at the same time, we need to meet all the regulatory requirements that could be different in each country. So, if we summarize all this in one sentence, in a clinical lab, we have to get the result out quickly, but without compromising quality. So we know this is complex. Now, before introducing a new test in the lab, we need to carefully design and clinically validate the assay. And to design a new test, there are several aspects we need to keep in mind. First of all, the clinical question, because it is going to determine the panel design, the sample processing protocols, how we are going to process the sample, the instrument setting, how the instrument, the machine is going to be set up, the data acquisition parameters, and data analysis strategy. So once we come up with the antibody panel, we need to test the entire assay. And this is an iterative process that can take many rounds of design and redesign before we complete it. After we are done with this testing phase, we need to ensure that our assay complies with the current in vitro diagnostic regulation, for example, the new IVD directive. So we need to put together a fit-for-purpose validation plan that confirms that the performance characteristics of our assay are acceptable for its intended use. So for that, we need to assess several parameters like the specificity, precision, sensitivity, which is essential for minimal residual disease test, stability, carryover, and even establishing reference ranges. But what happened at this moment? That there is no specific guidelines for validating flow cytometry tests in regulated environments. So the question is there a way we can get consistent result over time across instruments and operators, save time and save money while we comply with regulatory bodies? The answer is yes, by standardizing all the steps in our clinical routine. And that's exactly what the EuroFlow method is about. The EuroFlow Group is an independent scientific foundation made up of over 20 companies and research groups, which was born in 2006 with the mission of developing, standardizing, and validation of highly sensitive flow cytometry tests for diagnosis and follow-up. The EuroFlow method is a screening approach looking to use less antibodies at first to narrow down malignancy, those are the screening step, and then running the classification panels for further differentiation. They started with leukemia and lymphoma panels, followed by minimal residual disease panels to monitor patients. Once they got this system in place for onco-hematology, they apply all their expertise and knowledge for primary immunodeficiencies and also for immune monitoring fields. This method can be used in onco-hematology, clinical trials, and clinical immunology areas, but today we are going to focus on onco-hematology. So these methods include the standard operating protocols or SOPs, the antibody panels, analysis software tools, and a QA program. So let's go over each of them. There is a protocol for bone marrow sample collection and transport, specifically for multiple myeloma MRD testing, but also there are guidelines on sample collection for regular testing. EuroFlow also provides a protocol for sample processing and staining, including detailed explanation about extracellular and intracellular or cytoplasmic antibody staining, and other more specific, for example, regarding the nuclear TDT marker. There is also a specific bulk lysis protocol for MRD testing, and in those cases that we need to concentrate the sample. Just to remember that all this information is available online. For building leukemia, lymphoma, and myeloma panels, EuroFlow tests a large number of fluorochromes and antibody clones, so they select the fluorochrome based on the median fluorescent intensity, the stain index, spillover, stability, and availability. Here, we have an example illustrating the comparison between two fluorochromes, APC87 and APC750, conjugated with the same antibody clone, the CD10, and tested in the same sample under the same conditions. So we see that the APC750 conjugate shows a brighter MFA, which provides a better resolution between the positive and the negative reference population, but not only the median fluorescent intensity. As mentioned, they also take into account the stain index, but also the spillover, as we can see here. Similarly, for selecting the markers, they looked at the antibody clones, what is the best conjugate, the number of combined markers in a single tube, and the amount of antibody and volume of staining. So they tested the performance of the marker combination together using software tools, and that's what we can see in these diagrams. So these are multidimensional plots showing cases of TOL in green, BCPOL in blue, and AML in red, stained with the EuroFlow acute leukemia screening panel. And in each plot, they left out one of the markers in order to study one by one its discrimination power on these three acute leukemia subtypes. So as you can see, this result indicates that each individual marker included in this panel is essential, as missing one of them, cytoplasmic CD3, CD45, or cytoplasmic myeloperoxidase, affects the identification of the different acute leukemia entities. There are also protocols for instrument setup, calibration, and compensation available for different instruments. And these protocols cover the instrument configuration, that is, the setup of forward side scatter, the setup of the photomultiplier voltages, the compensation settings, and of course, the instrument performance monitoring. For our data analysis, we need flexible and advanced tools that make the multicolor analysis easier, for example, by turning off the background to just focus on the target population, and by looking at our data in many different ways, as you can see in these examples. Also, we need tools to simplify the multiparametric analysis. As flow cytometry technology evolves, there are more and more parameters to analyze. So to shape an idea, here we have all the possible combination of markers in an 8-color panel, that is 64 diagram. So instead of looking at all these to the dot plot, we can convert all of them into one multidimensional diagram that separates out population using all the parameters in our panel. So in this way, we can discriminate population with a very similar phenotype that can go unnoticed if we just look at a 2D dot plot. We can also check the complete immunophenotype of a target population in comparison with a normal control, or we can follow up patients with confidence by loading the diagnostic population onto the MRD case as a reference and see if we have a relapse or not. Using advanced tools, we can analyze patient cases, not single tubes, as we can combine all tubes from the panel, analyze them together, and create one single report. Still, the problem in flow cytometry analysis is the lack of a good reference point or baseline. As complexity of flow panels and data analysis increases, we really need big data analysis approaches. We need automation for our clinical flow analysis. So to put that simply, the automation consists of clustering all the events in the file, followed by an identification of all these clusters. But to be able to give to the population a clinical and a biological meaning to characterize this population and to interpret the results, we need to feed this algorithm with human knowledge and expertise, and that's the EuroFlow reference databases. So, well, let's see an example of automation in acute leukemia screening. So with it, with the automation, we get all the cell clusters, all the cell subjects from our sample identified and characterized. And after this, we get a compass diagram showing the cell lineage, TB or myeloid, or if it is mixed, that the aberrant cells are more likely to. This is done through a multiparametric comparison of your sample versus a reference database that contains B, T, and myeloid leukemic cells. And here we have the interpretation of the case, included information not only about the blast, but also the detection of potential related disorder of the patient. So you have the complete immunophenotype of each of the aberrant population, even if you have several lineage affected. Also additional information on the presence or missing population of the patient, sorry, because of the arrow. And most importantly, which panel to run next for further classification of the disease. Now, let's see an example of automation by in this case, for multiple myeloma MRD. So, after automation, we identify and characterize both normal plasma cells and abnormal plasma cells in red. And here we have the interpretation of the case, included a complete immune profiling of the patient versus a a normal reference, sorry, that appears here. Information on the bone marrow regeneration, the minimal residual disease level and the immunophenotype of the aberrant population, the clinical threshold that determines the sensitivity of the test, so we take a look on the limit of detection and lower limit of quantification, and information of the sample quality issues like hemodilution, taking into account the percentage and the frequency of mast cells, and also the sample viability, taking a look on the percentage of debris. Last but not least, EuroFlow runs two external quality assessment programs to certify the consistency and reliability of results generated in the lab. So the LSTQA monitors the overall flow testing performance from the pre-analytical to the post-analytical phase, including the sample preparation, the instrument settings, and the data acquisition. And in the case of the MMMRD QA, this QA specifically targets the data analysis of the myeloma MRD test. Therefore, the EuroFlow goal is to provide a standardized and validated highly sensitive flow cytometry test for diagnosis and follow-up of patients, and that requires a close collaboration between the industry and academia. Thanks to this collaboration, Cytognos can bring the EuroFlow method as a ready-to-use flow solution for any lab in the world. And that is what we call next generation flow solutions, and that includes the laboratory SOPs or standard operating protocols, the dry down antibody kits, the Omnisite instrument, the Infinicit software package, and ongoing training and support to help you along the way. So to wrap up, these are the key takeaways from the webinar. Flow cytometry is a powerful tool, a versatile tool in clinical practice, but it is still complex. However, the standardization is the way to obtain reliable and reproducible results. And this can be obtained following EuroFlow, which method is available through Cytognos as a ready-to-use solution for your lab. As always, if you have any question or you want more information about our next generation flow solution or any other topic, feel free to contact us. Here you have the contact of support from Cytognos, but also here you can find more information about the contact details to contact with Samu from BIOCOM Africa and with me, Arantxa from Cytognos. So if you have any question, query or whatever, just contact us without any doubt. And well, thank you so much for watching, for attending to this webinar. Now it's time for question or feedback, any comment that you could have. I am going to open the chat box and see if you have any question during the webinar. Similarly, maybe you don't have now any question, but later on, if you have any doubt about anything, you can write us and we will be glad to help you and to answer you. Okay. So thank you. Don't be shy. I mean, you can ask, but here you have the information, and of course, as I mentioned, here you have our contact details, but here this is a more generic, let's say, a email, so you can contact if you want more information about any of these things here, about the EuroFlow databases, Infinisite, also the academy section. And well, in the events area of our web page, you could find also new webinars that are going to be I think next week, so and more, so you can also visit our webpage just to be updated. So thank you very much. I hope that you find the webinar informative and helpful, and have a nice day. Bye.